Artificial intelligence is a game changer of the drug discovery process. So just a quick overview. Today it is so scientists sitting together and they have an idea how to cure a disease. And this idea has then to be tested in several experiments. And as it is, you have no prediction of the clinical outcome at the moment. So the idea is, instead of scientists, we are using artificial intelligence. So we take all the available clinical data, which is already existing, and then we look for biomarkers for better stratification in clinical trials. And on the other hand, we look for clinical verified drug targets. So that is the question now, okay, why is this not common in the pharmaceutical industry? And here I would like to hand over to my colleague.
Yeah, hi, quick intro. I'm Jörn Klinger and I'm responsible for the data science part at Beroceutica. So the question is, why don't all pharma companies use machine learning in this environment? So we have a little thought experiment here. Just imagine you have patients walking into a room and this is what's happening. Healthy, sick, and imagine you have about 1 million patients walking into the room, so the pattern is really easily to be detected. The rule is red shirt, sick, blue shirt, healthy. But unfortunately, in clinical trials and in drug discoveries, the data sets don't look like that. They look quite different, more like this, where you have very few patients, only about 800, a few hundreds, and you have a gigantic amount of features. So it would more look like this. So you have 800 of these guys walking into the room. Can anyone tell me the pattern? What determines whether one is sick or not? No, humans cannot do that, and most machine learning algorithms cannot do that either, but we have found a way to do that. So we detect patterns in data sets with very few patients and a large amount of features. Yeah, so here's just in the middle, you can see a summary of the results of all the tests we've run, both on simulated data and on real data. And what we can see here is that our method is really able to detect these patterns, even complex ones, polygenic ones, where more than one gene interacts with each other, much better than industry standards and standardized methods like Plink, for example. So, use case. How is this useful? For example, we've already used this to stratify patients with late-onset Alzheimer's disease, and our polygenic biomarkers found by our approach are able to give a more fine-grained and a higher accuracy of classification of these patients. How does this help the pharma industry? Well, if you do a clinical trial for an Alzheimer's medication, you first of all want to make sure that the patients you use in the trial actually have Alzheimer's and not some other kind of dementia. So this is where we can help there.
Last word on how we want to make money. The point already is that if you increase the success rate in a clinical trial, then you can really save a lot of money. And this is important also for, especially for small biotech companies, only use a few compounds in their pipeline, or small pharmaceutical companies. And as you see here, if you just increase the success rate in the phase 2, from 29% to almost 40%, then you can save up to $15 million per molecule per year. Only if you have a biomarker for a better certification, or if you have a clinical verified drug target. This is important, a drug target where you really know the connection to the disease state. Last but not least, I would like to say that we are here, we are looking for collaborations. So as you see, our USP is that we specialized artificial intelligence for clinical data sets. And we are looking for projects, collaborations with biotech companies, CROs, pharmaceutical companies. And that's it already. I only can say thank you for your attention and good luck.
Thank you.
Thank you for the presentation. I'm curious to know how you make sure that the quality of the data that you train on is sufficient, is good enough, because even big samples have a lot of noise, and even an AI maybe can sort of be caught in. And having that issue when you work in a black box setting, right? How do you then deconvolute sort of if something went wrong, how do you address that problem?
Yeah, seems like a good question for me. So first of all, the data sources that we have are genome-wide association studies. So these are studies funded by big pharma government agencies. So there's really a lot of money put into them, and they're conducted at the leading research hospitals across the world. So, and the data, of course, includes genetic data as well as clinical data. And as you can already see by the institutions involved, this data has a lot of money involved in it, and so it has quite high quality standards. Obviously, we also do our own quality control that starts with simple things, checking whether the SNP data of a single patient, whether a lot of SNPs are missing. If a certain amount is missing, we remove that. We look at the Hardy-Weinberg equilibrium, and really simple things like, does the gender of the person that is named in the study match up with the genetic gender that we can detect? And then we exclude data that doesn't seem to fit our quality standards. So far, I have to say that I was pleasantly surprised by the quality of the data we've seen. And then, other data sources would be data from pharma companies who obviously also hold themselves to high standards.
Could you flick to your data slide where you compared yourself to Plink? So is that the key slide that will convince partners to work with you? I'm just wondering, there are lots of AI companies around now, and the differentiation around proprietary access to data is one piece, business model is another, and value, what you're proposing is another. Is this key performance criteria the core of what you're offering?
Seems like another one for me. So, I mean, our main USP is that we are able to detect signal in these really difficult data sets with very few patients, huge amounts of features. And if you imagine you have already 1 million features and each feature is one SNP, then if you take interactions between SNPs, you really have billions or trillions of different features. And that also sort of expands our USP to being able to find these polygenic biomarkers where you have interactions of several SNPs. And this slide demonstrates that our method is actually doing quite well at detecting interactions between SNPs in such data sets, be they simulated or real data sets, whereas the industry standard is not. In finding customers, we usually take a data set specific to an indication and do out-of-sample prediction, and then we can tell them, look, we got your breast cancer data set here, and we can predict new patients at a 85 to 90% accuracy, and that is usually pretty nice when you can say that.
Well, I'm going to have a follow-up question on this one. What's the data behind this graph, which is the key graph, the key claim? I mean, what's the variability, the number of studies that you perform to support that result?
Oh, okay. Yeah, me again, always me. Well, basically, we started out simulating data where there were large interactions between SNPs. And we intentionally simulated the data in a way that it becomes very, very difficult for machine learning algorithms to pick up the signal. So we would create data where there are very few marginal effects, so very, very little effects that are just based on one SNP, but the effects are entirely due to interactions. So it becomes hard to detect that. We wanted to make it hard for our algorithm. And then also we used a combination of simulated data and real data. And then we used entirely real data and tried to find the interactions in that. And this is sort of an aggregate of all the tests we have run. I agree it would be more informative to look at every single test, but the take-home message is pretty much this, that Plink doesn't work well with interactions, and our approach does.
Well, in my case, that's an area which is very new to me to some extent. But I also feel that there are many companies working in the artificial intelligence also and analyzing all the data. So how many competitors are there in development to date, and how do you differentiate from them?
Complicated question. So the USP of the company is that we have another mass model behind what you have seen. And this is the USP of the company, and in the end, I think quality will win. So as you see here, we have to run with good results in the data analysis. So most of the data sets are free available, so we can compare with our competitors, our results. And This is our differentiation, the USP of our algorithm.
Are your competitors also using the same data source, or is it specific to you?
I mean, I think what we've seen when we did an analysis of the market and the competitors is that a lot of people use these buzzwords, you know, AI, machine learning, in biotech, in pharma, drug development, and so on, and they do approaches that look superficially quite similar, but when you dig a little bit deeper, they do things quite differently. So a lot of people are focused more on natural language processing. We have a small natural language processing component, but some companies really base everything on that. And so far, I would say we have not identified a competitor who does what we do in the same way with exactly the same focus and the same USP.
So how far do you think this is going to go? So if you can use your data sets to find new hypotheses for drugs, you can use another approach to find the right patients for run an in silico trial. When does the biology come in?
Sorry, what do you mean?
So when do you need to test in a person? Or will we get to the point where you don't really need to do any cells or mice or anything, and then you just do a clinical trial with 10 people, get your regulatory approval?
I would like to say it's complicated because you have to define your frontier. You know what I mean? So, for example, here with our Alzheimer example, we just do it only for stratification in clinical trials. So at the moment, you have here only ApoE4 as a predictor. This has an accuracy of 65%. And if you use our method, then you have almost 90%. So this is just only, we provide the data analysis of existing data for a better clinical trial. So I not really understand your question now, what you mean when it comes to biology.
I'm just wondering about the insights that you get from these associations and connections. And
Okay, no, I get it. So, and this is a little bit more the interesting part of here because, as I said, we have found here a so-called gene X. This is a kind of, we call it in-house safer, a rescue gene. This is a little bit unscientific, but okay. We looked a little bit more into the pathway, and indeed, it is a drug target. So it can be targeted by a drug molecule and has a therapeutic potential. But this has to be proven, and this is a little bit more in the future. So that's why.
Yeah, maybe just to sum that up, our whole process can sort of be divided in two. First is this discovery of biomarkers on a statistical level, but then the second element of our platform is really using contextual data, using natural language processing to do pathway analysis and to get to the biochemical reality of what's happening here, which is what we are working on and will be working on the third and fourth quarter of the year.
Following up on that, would you then be able to look at on-target, off-target toxicity also? I mean, that would be a nice step to skip also. Trials.
Off-target effects we can only see. This is difficult because it depends on the molecule. So here we are only looking at the drug target, so only on the pathway. We only give a statistical prediction of how good it is to go for this drug target. So what you mean, you have already then a ligand, a small molecule, an antibody, whatever, and then you have to do your phase 1 trial at least. But this we cannot predict. We are coming later, phase 2B or phase 3.
Could you tell me what, for your pathway analysis, it seems that, what's the purpose are you using? What tool are you using? And to what extent, essentially?
You're asking for the purpose of the signal pathway analysis? Okay.
Yes. Are you using commercial tools? Are you using your own pathway analysis? And is it just for the purpose to say, oh, this one seems like a druggable target? So industry wants to know, is your go, no-go policy for choosing drug targets the same as mine?
The key is here that we have to find already the interactions and associations in the beginning. So I would like to say the second part is a little more easier because you need at least the idea what kind of genes or gene expression profiles are important for the disease. And then you can use the standard techniques to predict the signaling pathways.
Also, so now I mean, essentially people sometimes use pathway analysis. If you analyze work Geschwind or thought leaders about this is to identify hubs or something like that. Are you using your own pathway analysis, such as KEGG, or are you using private tools, and are you actually using no hubs as a way of identifying your drug target?
I have to admit at this point, because we are here in a very preliminary stage, so we are focusing more here in the biomarker discovery at the moment. So the second part, you're right, we are now looking for all these available data sets or databases and try this, and we are working on our own solution in the future.
Thank you very much. Give it up, a warm applause for Beroceutica.